we'll move on to our next speaker, and that's going to be Dr. Corley Holbrook. Dr. Holbrook, you can begin sharing your screen. And Dr. Holbrook is a research leader at the USDA ARS Crop Genetics and Breeding Research Unit in Tifton. He has published over 500 technical publications and delivered numerous invited presentations at regional, national, and international meetings. He has served on the board of directors of the American Peanut Research and Education Society and the Crop Society of America. He was also the co-chair of the Research Steering Committee for the Peanut Genomic Initiative that was released in the last couple of years. Dr. Holbrook's talk is going to be breeding for pre-harvest aflatoxin resistance. So Dr. Holbrook, looks like you're ready and you may begin.
Thank you, Johnny, and thank you to you and everybody else that worked so hard to make sure that we could have a 52nd annual meeting of APRES. We'd like to talk today about breeding peanut for drought tolerance and reduced aflatoxin contamination. And I'm going to start my story back in the early 90s. The peanut industry, actually the Peanut Foundation, took the lead and approached the National Corn Group and the tree nut groups and the cotton groups, and they all joined together and lobbied Congress for an increased funding for aflatoxin in all those crops. They had an annual meeting, the multi-crop aflatoxin workshop, which was great because you got ideas from other crops. They had a pool of money that they, asked for proposals every year. And kind of back to Marshall's plug, they kept this going for about 20 years. They were in it for the long term. It was not their idea to end it. And I think there was a lot of progress that was made. And I agree with Marshall that if we're gonna, I've been banging my head against this wall for almost 30 years and we've made some cracks, but the wall's still standing. But if we're gonna take this wall down, we need sustained long-term research. So anyway, they were interested in a conventional breeding for reduced pre-harvest aflatoxin contamination in sound mature kernels. So I thought about it and I was developing a proposal. And I thought, what do you need to have a breeding program? Well, you need two things. You got to have genetic variation. You got to have genes that would give you reduced aflatoxin. And then you have to have accurate and reliable screening techniques that will enable you to identify those plants that contain those genes. So if you have those two things, you throw in some money and some time, you can make progress. Well, let me back up. So, oops, I didn't back up. When we started in the early 90s, we didn't have any of that. There was no indication there was any genetic variation for pre-harvest aflatoxin contamination. There was some indication that, there was some clear indication that we had genetic differences for colonization in vitro, but it didn't affect aflatoxin contamination in the field. And we didn't have reliable and accurate techniques that we could use at the large scale that you'd need for a breeding program. So what we proposed in our first proposal was to develop screening techniques and use those screening techniques to try and identify sources of resistance. If we were successful in those first two objectives, then we would begin a breeding program. So just a few comments about development of screening techniques. This was, I think this was critical to our success. Elizabeth Wills was a postdoc with Dave Wilson. Previously, they had used, most of the inoculation was a spore suspension in water. So you did get a spike in the number of colony forming units per cubic centimeter of soil, but pretty rapidly those populations declined. So Elizabeth developed a technique where we grow the A. flavus and A. parasiticus on pasteurized cracked corn that's been treated with antibiotics to inhibit the growth of bacteria. Then we inoculate the peanuts, and then that provides a food source for the aspergillus so that we'll have an extended period of fungal pressure on the developing peanut pods. So we use that in all our tests, and I think it's been critical for reducing our variation. And variation is really bad in aflatoxin. So We have that. It was suggested to us that we go out to Yuma, Arizona to try a screening out in the desert of Arizona. Arizona is a desert, very hot, very dry. I guess the real concern at the time was it going to be too hot? And you can grow a good crop of peanuts in Arizona. In fact, they at the time, they grew a couple of thousand acres with flood irrigation. To test the temperature question, we put out some shade cloth and grew some peanut plots under shade cloth. We grew them without the shade cloth. We irrigated and then, with flood irrigation, and then stopped the irrigation. And we did not see any advantage in the shade cloth. I think the information in the literature on the upper limit on aflatoxin production, the upper temperature limit, is not correct. So that was a good thing, because putting up the shade cloth was a pain. So we didn't have to do that, but we had a lot of escapes. We had a lot of zeros in our plots, and we were just planting Flow Runner and Pronto. Everything should have had aflatoxin. Well, thinking about it, the situation in Yuma was a lot different than what we saw in the Southeast. In the Southeast, when you go into a drought, of course, the plants start to wilt. They wilt more. But then they stay. They're still alive. They're under intense stress, but they're still alive for a long period of time. In Arizona, when you cut the water off, it's like pulling those plants up and throwing them in a dryer. I mean, they very rapidly wilt, dry down, they're bone dry. So you don't have the at least 20 days of stress that you need to get aflatoxin contamination. So we proposed to put in some drip irrigation, and we just provided just a small amount of water to the taproot to keep those plants alive, so we would have an extended period of drought. Well, that worked great. I mean, we pretty much eliminated our escapes. Our CVs went down. You still have a big CV, but it's down compared to what it was. And we screened out there for a few years, but then we realized that our rankings of our genotypes were different out there than they were in the Southeast, which is expected. I mean, it's a radically different environment. So we backed off of screening out there and just focused on our screening in Tifton. This is the first drought study that we had working with Craig Queen. He had some small drought shelters that had moisture sensors, so they would cover the plots, when the ghettos started getting a little rain. We needed a bigger area. So I was thinking about putting wooden structures with plastic and everything. And my senior technician, he had more sense than to tell me that I was crazy. He just said, why don't we get in the truck and take a road trip? And he took me down to Atlas Greenhouse, and that's about 20 miles away. And they build greenhouse structures. And we talked to the owner, and he said, yeah, we can build you some greenhouses on skids. So that's what we did. And this is one of them being pulled by the tractors here. We've refined this so we can now pull it with one tractor. We can get 100 plots under that, 100 single row plots. So there you're getting to, a level that's more amenable to a breeding program. We started out with 5 shelters. We used 2 planting dates. Today we have 10 shelters. We use 1 planting date. So you can see the two tests in the foreground and the shelters in the background. These plots have been inoculated with Aspergillus flavus and parasiticus. 40 days before harvest, we'll move these shelters on top of the plots, anchor them down, leave them for 40 days, and then come back, harvest the plots, shell the plots, grind the samples, take a 100-gram subsample and analyze for aflatoxin. So now we're to the stage of, do we have genetic variation for resistance? We have a screening technique that I'm comfortable with. Well, the U.S. germplasm collection contains almost 9,000 different accessions. Analyzing for aflatoxin is extremely expensive. I don't do anything unless it has at least 5 replications. My preliminary screen has 5 reps. Anything that looks promising, I want to do 10 reps in 10 locations. I mean, in 2 locations. So, you know, it's just not feasible to look at 9,000 accessions. So we proposed in our original proposal, we proposed to select a core collection for peanut and to screen the core collection for pre-harvest aflatoxin contamination. Core collection that we selected has 831 accessions. And this is a cartoon illustration of a core collection. So here's the core collection with 831 accessions. And just for illustration here, I have 5 clusters. We took all the data that was available for the 9,000 accessions, used multivariate statistical analysis on all that data, and made clusters, not 5 clusters, we made hundreds of clusters. And then randomly sampled from each cluster 10% to go into the core collection. Now the theory is that these are all genetically similar, so the indicator accession, which is the random sample going into the core collection, if it shows susceptibility, most of the accessions are all in this cluster will also be susceptible. This would be the cluster that you'd be interested in because your indicator accession was resistance. Now we've tested this theory, and you can see this in the literature, with late leaf spot. We examined the entire collection for late leaf spot and also nematode resistance, and the theory does work. So and then no matter what, the core collection provided us with a subsample that was feasible to screen for aflatoxin contamination. Here's one of our shelters, what it looks like after probably 20 days of stress, maybe 30 days of stress. You can see the genetic variation there for drought tolerance. There's a lot of variation, as I've said before, and I'll keep saying it, but there's a lot of variation from shelter to shelter and from year to year. Now, from a breeding standpoint, if we're looking at 831 things, we got to compare over shelters and over years. And so to do that, I calculate a relative toxin, which is the entry mean toxin divided by the mean toxin for all the entries in that test. And what we're looking for is genotypes that show at least a 60% reduction. We do the same thing for yield, although it's not as critical for yield, but there is shelter to shelter, year to year variation. So we do the same thing and calculate a relative yield. And for yield, we want to select things that have at least a 20% advantage in yield. We went through all the core collection. We did a preliminary screen, anything that looked good. We did additional evaluations with at least 10 reps in at least 2 locations. These are the accessions that we selected as having a moderate level of resistance to aflatoxin. Now, I never expected to find immunity in the germplasm collection. I did expect to find genetic variation, and we did find genetic variation. We have these. We also did an interesting study with Keith Ruckers, actually was a graduate student with Craig Queen. He looked at some of our material for drought tolerance. And he found, and we selected this based on root size. And we found that the most drought intolerant genotype, here it is with the visual stress rating, flow runner is pretty good, really. And then we have 2 PIs, one that's numerically better stress and one that's statistically better or less stress. And we looked at those, and this is Keith's data. We looked at those for aflatoxin, and this is the prettiest aflatoxin data set I have. We can see this intolerant genotype here has significantly more aflatoxin than flow runner, which has significantly more aflatoxin than the drought tolerant genotype. So this is very convincing data that in South Georgia, drought tolerance gives you reduced aflatoxin. We looked at, we tried a lot of different things. And so here's some other sources of resistance that we have. So then we need to answer, you know, can we use these tools that we've developed to breed for reduced aflatoxin contamination? And I think the answer is yes, but it's not easy. So here's the breeding procedure that we used. We take the resistant things and cross them with high yield, because we've got to take the resistance to aflatoxin and combine it with yield. We then produce the F1s and F2s, single seed descent down to the F4, where we harvest individual plants. All that I said, I don't do anything without 5 reps. So we've got to spend a year increasing seed. getting us seed that we can do a 5-rep test. We then have to also increase seed of all these so that anything that looks good, we can advance to a test that has 10 reps in 2 locations. Well, here's the results. We've made progress. We've got families that are really good. We've got individuals within families that are really good. Cross 431 family was one of our best families here. This is the number of tests that we have evaluated these genotypes in. The relative yield, they have drought tolerance, good drought tolerance. I define drought tolerance as yield under drought stress. And then a few of them, some of them have good reductions in aflatoxin. Not all of them, but some of them. So that's very promising. We have other promising families too. C7616, some of you are probably familiar with that. It's a drought tolerant line that we developed. We've had a lot of testing on that line, and it does have drought tolerance, and it's been documented in Texas, Virginia, Alabama, Florida. It's a good standard for drought tolerance, and it has some reduction in aflatoxin. This line was used as one of the parents in the genomics initiative to develop a couple of different recombinant inbred line populations. Check my time. Okay. So C7616, kind of the standard peanut germplasm line with improved drought tolerance. This family was actually a cross of Georgia Green by a PI, one of the PIs from the core collection that we identified had reduced aflatoxin. But then the last thing I want to just briefly mention is, okay, so we have all this material. ICRSAT also has a bunch of material. I had an opportunity several years ago to spend some time with Sean Niggum, who had probably done most of the work at ICRSAT. And I said, Sean, why don't we exchange our best stuff? So he sent me his best stuff. I sent him my best stuff for aflatoxin. And the plan was we'll test it in Georgia. He was going to test it in India and arrange for it to be tested in Africa. Unfortunately, Dr. Niggum retired, and I've not seen any data out of India or Africa, but I've got a little data here I'm going to share with you from Georgia. So this is just some drought tolerant study, and these are the worst lines from the aflatoxin set, and these are the best. See, there's the 431 family. 321 family is also a very good family. These 2 genotypes are highlighted because in the next year of testing, they were consistent. They were amongst the worst. Once again, you have the 431 amongst the best. So this is just a summary of the best for yield and the best for aflatoxin from the aflatoxin set. So you can see here the number of tests that it's been in. Mean relative yield from 1.4 to 1.2. And then for toxin, these are the best. These are the number of tests that it's been in. So this has been in 6 tests. That's very good. This has been in 5 tests. And relative toxin. So this was 30% of the mean of all the tests, all the 6 tests. If anybody's interested, I'll be happy to share this information with them. Sometimes you want a standard for susceptibility. So I also picked out the worst genotypes for aflatoxin, and here you got one that's been in 3 different tests, and it's 7 times the mean of the test. And then the worst for yield also. So in conclusion, possible, but far from easy to breed peanuts for reduced aflatoxin contamination. In Georgia, there appears to be a strong association between drought tolerance and reduced aflatoxin. The aflatoxin set contains several genotypes with good drought tolerance and/or reduced pre-harvest aflatoxin contamination. And when feasible, drought tolerance and/or reduced pre-harvest aflatoxin contamination would be ideal traits for breeding with molecular markers. And some exciting work is going on there right now. In fact, Reagan Wiggins is talking either tomorrow or the next day in the student competition. He's looking at some markers for reduced colonization and contamination in vitro. It's a kind of a follow-up for the work that Walid Korani did when he was in Tifton. As I mentioned, the C7616 was used to develop 2 recombinant inbred live populations. Those populations have been extensively phenotyped for drought tolerance, and work is ongoing to try and associate that data set with the genetic markers so that hopefully in the near future we can begin marker-assisted selection for drought tolerance and reduced aflatoxin contamination, and that would be a real game changer. Johnny, if I have any time, I'll be happy to answer a question or two.
Okay, thank you, Dr. Holbrook. I do have one question, and just from my own point of view, could you just tell how close are the lines that you're talking about to something that would be commercially viable?
Yeah, so that's an excellent question, and that's been the reason we haven't knocked the wall down, is because the other breeders like yourself continue to make progress in yield, and you know we're a step or two behind you in yield. We've got some things that I think would reduce the number of years that aflatoxin is a problem, but if aflatoxin isn't a problem, you're not going to have equivalent yield to the top yielding varieties.
Okay, thank you. That's, you know, we in our drought tolerance program, we kind of find the same thing. When it's really good for drought, it's not necessarily good when it's not under drought.
Yeah.
Diane, there was a question asked, are there any varieties that you know of that have proven to be more resistant to aflatoxin that could replace the varieties that are not?
I think that's a Corley question, probably, or a Barry question.
Corley, do you know of any?
I'm not aware of any. One thing that is a bit of a challenge in peanut, and I think Diane would probably agree with this, is most peanut varieties are pretty drought tolerant. It's a challenge to get better than the current varieties.
Okay.
There's an association between drought tolerance and aflatoxin. Most of the common varieties out there are not the most susceptible things that you could grow.
Yeah. Okay. Next question, we're moving to Corley, I think. Corley, do you have any plans to release at least the germplasm line for the C431-1-7? What about this line precludes its use as a cultivar for dryland production?
Yeah, so that's a great question, and actually, I've shared C7616 with a lot of people, but I have never officially released that one. So it's on my list to release too. But then, yes, right after that, I would, you know, I'd be happy to share 431 with anybody, but I do need to get that documented and in the germplasm system. Now, to the second part of that question, you know, there wouldn't be anything to preclude it from being used in a dryland situation. We did evaluate it under irrigation and full fungicides and made the decision that we shouldn't release it as a cultivar for those types of production systems. But I did not extensively evaluate it under dryland conditions, so it may have a place there.
We've got a couple of follow-up questions for Corley. Since the peanut is naturally drought tolerant, what do you see as other physiological markers useful for screening in your breeding?
Well, really hoping that Reagan Wiggins is going to come up with something. She'll have a presentation tomorrow or the next day. It would be more of an internal factor within the kernel that may affect aflatoxin production. Don't know exactly what that is, but I think what we're going to need to do, and that's one of the reasons I wanted to develop that aflatoxin set, is I think we're going to have to take a little piece from here and a little piece from there, drought tolerance, some internal factors, maybe some whole integrity, put them all together to bump up the level of resistance. And I do want to share one quote. It's in my mind. I wish I would have said it first, but I didn't. We had a meeting several years ago to debate whether ARS should continue doing aflatoxin research. Corn and peanuts. I mean, it's been, like I said, it's like banging your head against a wall. Are we making any progress? Should we continue? Well, I made the argument, and other people did too, that we are making some progress. And somebody stood up and said, hey, we haven't solved cancer, but we're not quitting on that. So I thought it was a great quote.
Yeah, that is. That's a good one. Another kind of coming at it from a different angle, would you think there is a possibility to approach aflatoxin by working with A. flavus in making it non-toxic? Kind of like the screwworm program, screwworm eradication program that was done back in the 1950s?
Well, Dawson has done that, you know. Aflagard is that.
Okay.
So it's just a matter of the economics, you know, will people pay to use it?